Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions from the window. Your questions will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Andrew Lees from Fina Bio Solutions.
Thank you for the introduction, Leah. In my talk, I will be discussing the development of a purification process of a recombinant protein, which is used in so-called conjugate vaccines. The protein has applications in both clinical and veterinary fields. I'll first tell you a bit about Fina Bio and also about conjugate vaccines, so you have that background. Fina Bio Solutions' core area is in conjugate vaccines. Conjugate vaccines are vaccines which need to be chemically assembled and include vaccines for strep pneumonia and meningococcal disease. These conjugate vaccines are among the most complex and expensive of routine childhood vaccines. Part of our mission is helping to make these vaccines more affordable for low-income countries. My specialty area is in the chemistry of conjugate vaccines, and much of our expression and purification work is geared towards reducing the cost of the protein component of conjugate vaccines. So what are conjugate vaccines? There are many antigens which are poorly immunogenic unless chemically linked to a protein, which provides needed T and B cell epitopes. These antigens include oligo and polysaccharides, peptides, chemicals, and even some proteins. Chemically linking, for example, a peptide to a protein, the carrier, creates the peptide protein conjugate vaccine. There are only a limited number of carrier proteins which are approved for clinical use. These include tetanus and diphtheria toxoids, which are toxins that have been chemically detoxified. A third widely used carrier protein is CRM197, or CRM, a genetically detoxified diphtheria toxin. CRM has been too expensive in general to use in low-cost vaccines. To address this, Fina Bio has developed an E. coli strain with an oxidative environment which can express disulfide bonded proteins like CRM197. It expresses them as soluble, properly folded intracellular proteins. And unlike comparable commercial strains, our E. coli is not crippled and can grow to greater than 300 grams per liter. This has allowed us to markedly reduce the cost of the protein. There's also a need for additional carrier proteins in order to prevent carrier protein overload and the fact that some antigens just seem to work better with one carrier protein than another. We have therefore used our E. coli strain to develop a new carrier protein, a tetanus fragment, tetanus toxin heavy chain fragment C, or ttHc. And I'll be discussing the purification of this protein and our efforts to make it more affordable. Our target protein, ttHc, runs as a 50 kilodalton doublet on STS page. It's slightly acidic, expressed as an intracellular protein, so our feedstock is an E. coli lysate, and it's expressed at greater than 1 gram per liter in the fermentation broth. Our goal was to produce it at greater than 95% purity with low endotoxin and host cell protein, and we wanted to be able to scale up to multi-gram levels. Our primary analytical method is STS page. The center lane here is our feedstock, and on the right is some purified ttHc, which we had previously prepared. In developing our purification for TTHc, we first considered the suitability of various modes of chromatography such as anion-exchange, HIC, etc., based on literature, experience, and some early static screening experiments. In our case, we found that anion-exchange and HIC were most suitable. We then selected candidate resins in 1 ml pre-pack format and screened them using long, shallow gradients. We then selected a limited number of resins to further evaluate for their binding capacity and selectivity, and then to eventually convert the linear gradient to a simpler step gradient. As the first step, we reviewed strong anion exchangers for capture. For this study, we considered only Bio-Rad resins with varying properties. The Macro-Prep high Q is a relatively small bead with a high charge density. It's roughly twice that of Cephirose Q, and it's built on a methacrylate-based resin. The Nuvia resins are so-called Cas beads synthesized from water-soluble acrylamido and vinylic monomers and directly incorporate the ligand as part of the synthesis. The Nuvia HPQ is a smaller bead with a relatively low charge density. Low charge densities are often great for more selectively binding very large molecules like IgM, which can bridge ligands. The Nuvia Q resin is a larger bead and also has a surface extender, which we have found helpful for capture steps. So we looked at these three resins. We used 1 ml 4-site columns from Bio-Rad with a 20 CV gradient to 1 molar sodium chloride. Each had a reasonably good binding of the TTHc protein. Shown on the left is the Macro-Prep, in the middle the HPQ, and the Nuvia. With these long gradients, neither seem to have very high selectivity, and although initially the Macro-Prep Q looked like it might be the better resin, on further study, looking particularly at binding capacity, we found that the Macro-Prep Q had much lower capacity. We then went on to look at compare more directly the HPQ and the Nuvia Q. In a further study, we determined that the Nuvia Q had almost twice the binding capacity of the Nuvia HPQ. This may be a reflection of the lower charge density of the HPQ versus the Q. We found that the Nuvia Q resin had a dynamic binding capacity of about 10 milligrams TTHc per mil of resin. This may sound low in comparison with antibody binding capacity, but keep in mind that here we're loading whole filtered E. coli lysate right onto the column. There are a lot of competing substances. We found that the binding capacity was unchanged up to at least 375 centimeters per hour. In order to improve the purity profile, we ran a shorter, shallower gradient and found that the TTHc eluted relatively early, with very little of the target protein coming off in the high salt 1 molar strip. We then converted this gradient to a step elution of 0.15 and 0.4 molar sodium chloride, this time also scaling up from the 1 ml to the 5 ml columns. And here you can see that at 0.15 molar elution, we achieved about 90% purity early in the gradient. We then took this eluent and moved on to an orthogonal binding step, looking at HIC binding. Bio-Rad has two resins for HIC, Macro-Prep T-butyl and Macro-Prep Methyl. Again, these are based on their methacrylate resins. We found very low binding capacity on the on the butyl and good binding on the methyl. This seemed a little unusual. I had expected to have higher binding to the butyl, but on looking at the resins in more detail, the butyl resin we found has a carboxyl groups as well as the butyl groups. So in fact, it is a mixed mode resin with almost the same cation exchange capacity as many of their S resins. As both the protein and the resin are negatively charged, the protein being acidic, it wasn't surprising that it really didn't bind. In contrast, we had good binding to the methyl resin. You'll notice here also that, in fact, these are not really strictly only hydrophobic resins. Even here, there are other interactions in the ester linkage of the methyl group and with the additional hydroxyl group. So really, even though these are called HIC resins, they really both have elements of mixed mode chromatography. So the anion exchange eluent was directly loaded onto the methyl HIC after adjusting the ammonium sulfate. The target protein eluted early in the gradient. So we'd say it bound relatively weakly. In trying to achieve higher levels of binding, we found that this protein started to precipitate when we reached 1.5 molar ammonium sulfate. However, the eluent from this column was quite pure, and there was very little target protein in the flow through at this loading level. So we converted the HIC column to a step gradient, initially finding that it began to elute about 90% and completely eluted at 80% of the buffer B, which was 1.4 molar ammonium sulfate. So it really eluted quite early in the gradient. We then had our purification process, harvest and lyse involving centrifugation, our capture anion exchange, and followed by HIC. This was the final product of the combination of Nuvia Q and Macro-Prep Methyl. And we more than hit our target. We achieved better than 99% purity by SDS Page and SEC HPLC with low endotoxin and low host cell protein. So our next steps are to scale this up by increasing the column volume and to further optimize the HIC column. In particular, we would really like to increase the binding capacity of the column, and we will be evaluating other resins. I'll now leap to a conjugate vaccine application using TTHc as the carrier protein. Cattle tick fever disease is a major problem across the world. It's caused by the parasite bovine babesiosis and spread by those nasty little ticks. While once widespread in the US, it's now largely confined to the southern border, where it could cost the cattle industry up to $1 billion a year. There's currently no vaccine for the disease, and control is addressed with tick control, limiting cattle movement, and culling of the herd. Fina Bio has been working with researchers at Texas A&M University who have developed a peptide-based vaccine for cattle tick fever disease. For this peptide, TTHc seemed to be the best carrier protein. TTHc was derivatized and the vaccine peptide chemically linked to the protein. A field trial is now underway with results due in a few months. If successful, we expect to scale up our purification process to produce kilogram amounts of TTHc. Animal vaccines need to be very inexpensive, and we will be applying our expertise at making low-cost human vaccines to these veterinary vaccines. I would note, however, that unlike human vaccines, with a cattle vaccine, you can eat the recipient. With that, I would like to thank you and express my appreciation to Bio-Rad for their technical support and to the Fina Bio staff who performed the actual work that I talked about. And finally, I would like to thank my COVID companion, Miss Molly Mischief. And with that, I would be happy to take questions.
Okay, great. Thanks, Andrew. So the first question is, to make this a cost-effective vaccine, how do you approach downstream purification choices?
The main consideration is to keep the process as simple as possible. We want to minimize the number of steps, and we want to minimize the amount of processing between steps. So we want to be able to load directly from the eluent from one column onto the next column. And as you can tell from the process we developed, we went right from the eluent from the anion-exchange column right onto the HIC column. So that saved having to do any TFF steps. Other considerations are trying to achieve high loading capacity and high flow rates. And the newer generation resins, like the Nuvia, meet those requirements.
Okay, great. And why does Macro-Prep High Q provide lower dynamic binding capacity than the Nuvia Q when it is the highest measured ionic capacity?
Yeah, that's a good question, because that was a little surprising to me. I really thought maybe we'd get higher binding capacity, but on discussions with the Bio-Rad technical staff, it seems like the reason is that the newer resins, like the Nuvia, have a more optimized pore size. In the cartoon pictures, they often show pores as sort of these uniform holes going through the beads, but in fact, they're fairly tortuous, and many of them, in older resins especially, more or less dead end. They narrow and then they dead end. So the Nuvia resins have pores that have been more optimized, so there's actually more useful surface area that's available for binding. And I think that's the reason why the capacity is higher.
And would you consider using an anion-exchange hydrophobic-interaction mixed mode resin for a possible one-step purification?
We're definitely going to be looking at that. I like the concept because obviously one column would simplify things a lot. I would consider it highly unlikely that we would be able to get to a high enough purity with just one column, because we're loading whole E. coli lysate onto the first column. So it's unlikely, but it's possible, and especially for veterinary use, if we can get the endotoxin levels down, that's certainly a possibility.
Okay, and what is the difference between HPQ and Nuvia Q resins?
It's a good question. The HPQ is a smaller bead than the Nuvia Q, and it also has a much larger pore size. Combined with, as I had indicated, a much lower charge density. So what this means is that the HPQ may have higher resolution because it's a smaller bead, and it is better for binding larger molecules, where the large molecule can bridge the low charge density. This type of resin that has a low charge density is generally more suitable for these very big proteins. And the Nuvia Q is a larger bead, well suited for capture steps, and it has a higher charge density, which is more suitable for smaller proteins like the TTHc.
Okay, great. Well, it looks like we have time for just one more question. If you haven't asked your question or if your question hasn't been answered, just please go ahead and type your questions in. We'll be passing along all the questions to Andrew, and he'll be able to follow up with you directly. So don't hesitate to continue typing in the questions if you have more. The last question is, what makes CRM 197 so expensive?
Well, initially, the protein was only made in Corynebacter diphtheria, the bacteria that makes diphtheria toxin. And it's a fairly tricky bacteria to grow with some stringent, I think, iron requirements. And most people have only been able to get about 100 milligrams per liter of the CRM 197 from the Corynebacter cultures. Some companies have developed some high productivity strains, but they're also proprietary. Once it began to appear in other bacteria, notably in Pseudomonas, I would say it's as much a business decision to keep the price high. With Fina Bio solutions focus on making low-cost conjugate vaccines and promoting them in making them more available for low-income countries, as well as making them the protein more widely available for research and development of conjugate vaccines, we deliberately set the price as low as we could. Our E. coli strain is very productive. For the CRM 197, we make about 2 grams per liter, and we decided that we would set the price, as I said, as low as we could rather than as high as we could. It's perhaps a strange business philosophy not to make as much money as you can, but what we do as the only provider of CRM 197 who both provides the protein and has expertise in the area of conjugate vaccines, we leverage that to increase sales and to help promote the product.
Okay, great. Well, thank you, Andrew.
Thank you, Leah.
And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.